Hi, welcome on Dr. Microbe. Here we talk about microbes. And today we talk about microbes in Alaska. Oh no, you're not drunk. You're watching a French speaking English.
Bring it on.
Biology, and in particular microbiology, requires samples, like a lot of scientific domain. Sometimes those samples are very easy to get. You just have to bend over. Sometimes they require a little bit more organization. Like today's samples that I went to collect in Alaska. So yes, a bit more organization. Because today we are talking about how I went on over teams of scientists in the middle of the Gulf of Alaska to gather samples. I will tell you the story of how scientists play with muds. To gather samples, we need to do a scientific cruise. But what is a scientific cruise? Well, it's not a cruise like, old people emitting way too much greenhouse gas while drinking margarita in the middle of some random sea. This is like some useful cruise. Bunch of scientists on the boat, gathering together, getting samples, coring for mud on water samples. The usual stuff that you kids do too when they're at the beach, but this time it's made by professionals. It's not very different. And yes, today I'm talking about an adventure that will take me from Los Angeles in California to the middle of the Gulf of Alaska. We will go by Anchorage on SeaWorld. You will not see my face a lot because you will have a lot of record of beautiful landscape, like that. Or that. Or even that. But first, being all mud and giggle, let's go and do the important stuff. This video has been written by me. It's my own word, my own feeling, my own feedback. Nobody forced me to write anything, neither the Sequoia crew or the many teams of scientists that were with me on the boat, and it's not representative of the rest of the crew or any of those other scientific on the cruise. It's just me and my opinion, on my very French accent. How did I go from LA to, cold? It's very simple. You take the flight to LA and you end up in Anchorage. From Anchorage to SeaWorld, you become the journey of 2 hours in car. But it's not like 2 hours of random journey. It's like very nice drive, as you can see on the footage. And then we arrive at SeaWorld after driving for 2 hours in a very beautiful landscape. We arrive at the place of embarkation and we start loading our luggages on the rest of our equipment. We get to know the other researchers who are with us, because yes, it's a big cruise, there are several labs. We are professional here. Kind of. And we walk in SeaWorld, a very small village in Alaska, very nice, that I can see being a beautiful backdrop for a book by Stephen King. By night there is a strange fog that kidnaps people, or there is a weird cult story, and it's written by Stephen King. But it's a pretty place. Honestly, it's a very pretty place. But I think I read too many books. It is there that the Sequoia is located, the boat that will be the living place of several dozen scientists, from technician to principal investigator, or PI, which is like the technical term for boss researcher. the researchers who are the boss. Oh, yeah. I hope you like sharing a place to live and, having roommates, because for 2 weeks you will have roommates. Let's talk about the Sea-Kuliak. The Sea-Kuliak is owned by the National Science Foundation, or the NSF, a foundation that was pretty useful before it got, well, fucked over by an orange, ugly goblin. Because science sucks, apparently, and it's operated by the Alaska Fairbanks College of Fisheries and Ocean Science. It's a nice boat. It's a very nice boat. And then you socialize, because there will be a lot of people to work with, so it will be nice to be a little bit polite and civilized. For 2 weeks, you better get along well. And yeah, and by the way, this video will start as like the first 3 days of how we started, then we will be a bit more broad and general. And you will see. And by cutting it into days at the beginning, I can also show you like what a scientific cruise is during the day or during the night. Yes, because you will see there is a bit of a different rhythm. For example, we divide our small team of researchers on the boat crew into 2 teams. The day team, who work during the day, and the night team, who work during the night. Yes, thank you for following. And you lucky bastard, you're going to have the point of view of the night shift. The best shift. Obviously. The best shift. On the schedule, a shift that starts at midnight and ends at noon. Believe me, coffee is your most valued ally. The second day was not the most hectic for most researchers. We had the security meeting to find out what happened if the boat sinks. It didn't sink, but safety is important, And we had an update on the different danger posed by a boat. If you are a sailor, you know what I'm talking about. But for the others, a boat moves. It rocks on everything that is not attached also moves, including you. So we are much more prone to an incident. In addition to that, we will sample with something called a CTD on the core. A CTD, well, it's the thing with the bottle on the screen. On the core, It's the other long thing that you see on the screen. And it has this constraint, as you guess, like the tensions that need to be precisely monitored so that the cable doesn't break. So it's a heavy machinery and it requires some precaution. And of course you need to be updated on those precautions, safety measure. Because the cable is under tension, so if it breaks, how I can say that? Well, I'm probably exaggerating, but let's just say, well, yes, probably. Safety. Anyway, everything to make you aware that the boat is a dangerous place, so we are being careful. Once again, safety first. Then we finally visit SeaWorld one last time, and we go to bed for our first day at sea. And that's it, our first day at sea. Ooh, exciting, isn't it? Well, yes and no, because on the first day it's a bit like Magic Up in Pokemon. Nothing happened, which is really practical because we have to finish getting ready. Solutions in bottles, tools, tubes, syringe, in short, it's like Breaking Bad, but legal.
Yes, science!
We need organization. We even have liquid nitrogen. It also allows us to realize if we are seasick or not. Because we're going to be out of town for a little while, it will be a shame to be sick in addition to working 12 hours a day or 12 hours a night. In short, you have the idea. It eases you into it. So yeah, that was the first 3 days. Let's be a bit more broad now. Let's talk about coring. What happened when you core? Well, that's an excellent question. Coring, it's delicate, especially when sampling at several hundred or even thousands of meters depth. It's a delicate process that involves heavy machinery and qualified people. And I'm neither of that. So we need people that can do that. But how do you know where to core? Well, in general, we either have preliminary studies or teams that have tried the field or that give us an idea of what awaits us. In other cases, we poke around and use different tools to measure the condition around us. On what tool are you asking me? Well, let me introduce you to the CTD. Basically, a sonar for depth on a CTD, a device that measures the conductivity, temperature, and density of water. Conductivity, temperature, and density. CTD. Yeah, the guy who created that had imagination. We also measure the salinity, oxygen, concentration, and tension. The tension of water? No. Tension of something else. We'll get to it a little bit later. The interest of the CTD is that it allows us to know where we are stepping, which is always pleasant in research, because this does not happen often. So we have an idea of the condition at the bottom of the Gulf, where we are. We can pair it with a rosette, which is a little device that you see on the screen and that is connected to a whole bunch of open-ended bottles. But what's the point? Well, we are going to throw that in water. Slowly, we are not monsters here, we are scientists. Monster with a degree. And we will follow the condition of the water according to the depth. In parallel, each bottle will be able to be closed individually during the descent. Slowly, we are not weirdos, we are researchers. Weirdos with a degree. Which means that if you have 10 bottles and sample your water to a depth of 1000 meters, you can open a bottle every 100 meters. The fact that it open at both ends will allow water to be sampled very easily without mixing the water in the water column. Sounds like a detail, but in science every detail can, bite you in the butt.
Details matter.
So let's be, detail conscious. In short, you put your rosette in the water. The descent is neither fast and furious. We are in for science. Everything takes time. From memory, we are on a descent of 10 meters per minute, which can take some time. In return, you have data on your water column, and so you can sample for a lot of things. At random microbes, because I'm a microbiologist, I'm a doctor in microbe. I spoke to you earlier about tension, because it doesn't seem like that, but all these devices pulling on the cable, a cable that may be very solid, but it's not because it's very solid that you're not careful. We therefore monitor the voltage carefully, and we also have a security protocol. We are not crazy either. We are not that crazy. When the CTD is in the water, either everyone is inside or it's mandatory to wear a helmet. It's like a motorcycle, without the sensation. Then the CTD will go down. We will follow the evolution of our different factor, sample, and it will go back up. And then that's where the fun starts, because we sampled water. Now let's see about mud. A core is a long tube of sediment that we extract from the ground. It's not a reserve for the marine environment. I did some during my PhD, for example, about a lake in Denmark. What we are going to do is send a tube several meters deep directly into the seafloor. With its own weight and inertia, the tube will sink and capture sediment. Then, with a mechanism at the bottom of the tube, this will hold back the mud. So, it doesn't escape during the ascension. So, basically, you send a tube down, it gets mud, there is a clap, there is a little trap at the end that holds the mud when it pulls back, and here we go, you have mud, if everything goes well. Sometimes it doesn't. Maybe it seems to you like theoretically it's very simple. And theoretically it is. It's a big pipe on mud. Can be very complicated. Except that we are in science. Everything can be complicated. Indeed, we talked about tension when we talked about the CTD. And the CTD remain relatively light. These are several liter bottles. They're not light, but they're not that heavy for the machinery. That heavy. I remind you that we are on a big boat, except that there we are talking about several meters of mud surrounded by a large steel of tube containing also a large tube of PVC. It's a bit heavier. It will therefore cause a little bit more tension. And once the tube rises, you will have some tension to it. So once again, the process of lowering a core takes at least an hour, after which you will get off the bridge to avoid any accident in case the cable breaks. Because safety first. Then you will put this huge mass of mud toward you. It will take, again, a few tens of minutes. So that's nice. Now it's time to work. Yeah, that was just the appetizer. Well, yes, when we put this kind of sample back together, we want it as fresh as possible. So what we do is we cut each carrot into smaller carrots, about 1 meter wide, if my memory is correct. I don't think my memory is correct, but deal with it. And those 1 meter core will be cut into smaller core, depending on what we need. For example, we have carrots that were cut every 10 centimeters. It's a bit of work, but we have to keep ourselves busy. We can never make simpler rounds of mud. For example, we can just cut the core into huge slices of mud. Looks a bit like cake, but please don't eat the mud. Or if we want to be a little bit more broad over a bit more depth, we can just be a bit less subtle and just take a drill and drill a hole in it. At this point, watch out, because those core are slightly pressurized, so you might have some leakage. So you drill your core, you have some sediment that you sample with your syringe for, porosity, pore water, microbiology, geochemistry. Well, you have a plan, so you should be good. For example, you take a sample to look at the density of your core, the different chemical species that are present in them. We can extract DNA to do sequencing, or RNA to see the activity of our microbes, or we can do fluorescence microscopy. I like fluorescence microscopy. I have several videos about fluorescence microscopy on the channel. I'm just saying. And if you don't know what fluorescence in situ hybridization is, you dive microbes depending on what they are. So you can have pink microbes, and yellow microbes, and green microbes. In short, the coring is a long and complicated process that requires the help of qualified people to make it go well. So please, in the comments, say thank you at least to the crew and all the technicians that help us to have very good quality sampling. Like, we couldn't have done it without them, so at least a thanks in the comments. But we don't have only big long core, we have also baby cores, or multi-core. It's a technique that allows to make several smaller core at once. That's when you're more interested in the 30 to 40 centimeter of your sediments. For the American watching that video, a centimeter is a real unit. It's not like your inches and stuff like that. Broadly, 30 centimeter, 1 foot. This technique has advantages. First, it's faster, and it also allows for good coverage of your sample since you have 5 or 6 identical carrots. But it's not very deep though. But as Greek philosopher Mick Jagger once said, the other advantage is that since multicores are smaller core, they are easy to sample. And you're in luck because I have a beautiful footage of us subsampling multicore core. In short, because the whole process took an hour and a half. But I will put it on the channel too, so you can see how long it takes. It takes a bit of time. How to do that? Well, we put the baby core on an extruder, we push, and thanks to the template of several centimeters high, you cut your segment with a non-precision. You can cut 5, 4, 3, 2, or even every centimeter if it's your fancy, which can be convenient. You cannot do that on a 6 meter long core. I know there is people who does that, but I want to keep my sanity. I want to keep the little sanity I have left. On board, we don't have only like core on CTD, we have our fancy piece of equipment such as a fridge. A container fridge. Indeed, we need to keep our core cool before cutting them. Well, we want to keep them as intact as possible before subsampling them. Since we don't wait to finish with one core before taking another, we can find ourselves being, overwhelmed with different core. For example, if your core at one site and your next site is an hour away, well, you're not waiting an hour or 2 hours that you finish your first core to sampling your second core. You sample as you go, which can lead to the situation of having several dozens of core waiting for you to be cut in a cold fridge. Yes, you will have work. We also had a device to scan our carrot with X-rays. It allows us to visualize the organization of the sedimentary layer in your core and to extract a whole bunch of data from it. I don't specify too much because I don't want to say any mistake and it's very not my field of specialty. In short, we have our small team that is going to travel the Gulf of Alaska for 2 weeks on your Urban 7 to hit the night shift. So on one side it means that I'm working from midnight to noon, but on the other side it means that I can see landscape like the one you see on the screen, which for me doesn't occur that much often, to be honest. Also it's the opportunity to see weird sunsets that just turn into a sunrise 2 minutes after, which is disturbing enough to be cool. So yeah, the other team saw bears and orcas, but it's okay. Night shift is cooler. Oh no, let's talk about what a day looks like. How is a typical shift looking when you are a researcher on a research vessel? Well, bad news, you don't have a typical shift because there is no 2 days that are alike. But in short, one evening you get up and you are in transit for, let's say, couple of hours. You can be 1 hour, 2 hours, it can be 16 hours. It means that it's the next group who will complete the coring and the cutting, which is a little bit fun because usually it's like the day shift, cut on samples, you arrive, you don't have anything to do because you're on transit, then you arrive on site and you go to bed on the day shift to do the coring and sampling. Well, the reverse happened too. And anyway, you still have something to do because it's not because the day shift started the coring that you will not take over after. Given the amount of cores you will have to do, do as you can because there is a lot. There is a lot. There is a lot of mud. I talked about time when you have 16 hours to, roll your thumb, but there is also time when you have 15 minutes to take your breakfast. Breakfast that is literally the most important part of your day. With mashed potatoes, bacon and eggs and half a dozen cup of coffee at least. Because there was days that were reasonably busy, so the good side is the day came by very fast. The bad side is you're exhausted and you need more coffee. And then, after your 12 hour of non-stop work, what do? Well, you eat first, because you have to keep up with it. And after, the mistake can be to just go crash in your bed and sleep too much. then not getting enough sleep because you're not tired, and then you get too much sleep, and then the cycle continues. Yep, been there, done that. Let's say that for safety, you get up at 11 p.m., so you have an hour to wake up, take a shower, eat, and up you go for playing with mud. You drink 1, 2, 3 cups of coffee, and then you take a whole thermos of it, just to be sure. And then again, you start again. In truth, it seems rough like it, but you get into the rhythm very quickly. Think of it like being in a music festival. You're exhausted, but there is so much cool stuff going on that you can keep going for days, on days, on days. And it's even better because, contrary to music festival, the food was good and the toilet were working. And yes, you have to adapt to the schedule, because you are on a boat to work all the time. You have only 15 days, not 16. And something happens all the time, and there is always a new call to cut, a landscape to discover, a sample to preserve. You're very quickly lost if you don't rest. And you know what's dangerous? That you don't feel tired, but you're on the boat. It's still dangerous, and if you're tired, you pay less attention. That's how accidents happen. I said it again, but safety first. An example? Well, one day I had 2 hours of sleep and stayed up 26 hours in a row. I felt fine. It was probably not good. So not really a typical day, which makes it all the more exhilarating. You lost track of time, you are settled by the core sample on breakfast, and you don't need anything else. And why do we do that? Well, for science.
Yes, science!
My role here as a postdoc was just to lend a hand, and in particular to help with the preparation of samples for fluorescence microscopy. Lost yourself on the channel, once again you will see we have several videos about fish. And if you like the content, by the way, just, put a like, let a comment, subscribe, so YouTube will recommend you more scientific content. It will grow my channel, and everyone will be happy. In the meantime, it will allow you to see when the next video will drop, which will be about a robot in the Gulf of Mexico. Yeah, don't miss this one. But in short, our little team was looking at microbes in the deep sediment and see if they were still active. Well, science takes time, so we still don't know, but we never know. Moral of the story, science is cool and needs to be funded. It needs to be funded to allow research ships to sail. It needs to be funded to allow teams to launch such projects. It needs to be funded to push back our knowledge on our small blue planet. Is a research cruise a vacation cruise, like you can hear people say time to time? Well, if you have 2 brain cells and those 2 brain cells are fighting for the third place, you might be tempted to say yes. But if you listen to this video a little bit carefully, you might be able to say
No, God! No, God, please, no! No! No!
a cruise is first organization, a good crew, passionate scientists, a team that help each other, and above all, extraordinary people to work with. Extremely hard to say. English. And so here I am back in Los Angeles, head full of souvenirs on mud. Mostly mud, but that's fine because we came from mud, so yeah, I get what I want. I saw magnificent landscape, meet wonderful people, and live a unique experience. So unique that 3 weeks later I boarded to another boat for another project. But that's the story for another video. Thank you for the Sea Kuliak and the crew to help us along this wonderful adventure. Thank you for all the different researchers I met that were all wonderful people and made me had a very good time for my first cruise. And thank you to you for watching this video. I hope you liked it, you find it entertaining, and you learn stuff. If you have, I don't know, cruise experience or research experience, please share it in the comment. I'm pretty sure people are interested with that. And me, well, I see you next time for more videos on more microbes.